In this video, I'm going to walk you through assembling and analyzing an Amplicon dataset from Ion Torrent using SeqManEngine. The dataset contains 28 Amplicons sequenced to a high depth of coverage, and I'll be assembling the data against the whole human genome as a template. So the first step after launching the program is to select your project type. So I'm going to keep the selection of genome assembly and click Next. And then it asks me for my assembly type. And I'm going to keep the selection of templated assembly large genome since we're using the human genome, and then click Next. Here I need to define my project name, so I'll go ahead and enter that. And I also need to define a project folder, which is where my output files will be saved. And I already have a folder set up for that purpose. Then I can click Next. And then I need to specify my template sequence. And for this, I'm going to add a genome template package, which is a package of data that we provide on our website for a variety of organisms. And it includes the template sequence, annotations, and associated dbSNP entries for that particular organism. And you can download these by clicking on this link here, which will take you to our website where you can select from the available data packages. But since I already have mine downloaded, I'm just going to select Add Genome Package and navigate to where I have it saved. And then I can click Next. And then I'm asked to specify my sequence files. So I'm going to select Ion Torrent from the Read Technology field. And then I can select the SFF file that I have from Ion Torrent containing my reads. And then I can click Next. And this page allows me to adjust the assembling parameters for my project, but I'm actually going to leave the default settings as they are because based on the project and assembly type and read technology we selected, SeqManEngine has already optimized these parameters for me specifically for a reference-guided assembly of a large genome using Ion Torrent data. So although I have the option to adjust any of these parameters, I'm going to leave them as they are. But one thing I do want to point out is under Advanced Assembly Options, if you look at the SNP tab, the default settings for SNP calculation is diploid Bayesian. And you also have the option to choose haploid if that's applicable to your project, or you can choose to do just a simple percentage calculation. I'm going to leave the value as diploid Bayesian, and then I can click OK and Next. And that's all that's necessary for setting up the project. The final step is to click Assemble, and SeqManEngine will begin assembling our data. Once the assembly is complete, it will automatically be opened in SeqMan Pro for analysis. The first thing you'll see here is the project window, which shows the contigs that were created from the assembly, as well as the length and the number of sequences within each contig. And you can sort this list of contigs if you wish by clicking on these column headers here. From here, the first thing I'm going to do is open up the Snip Report, and I can do that by making sure that Unlocated Contigs is selected, and then choose Snip Report. So notice that there are 145 SNPs found in this report, and I'm going to do some filtering here, but first I want to point out a few columns of interest. If you scroll over a bit, you'll see that there are 3 columns related to the Bayesian statistics that were used to calculate these SNPs. One is the genotype column. Another is the p0ref column, which shows the probability that the SNP is a call different from the reference sequence. And finally, there's the qcall column, which gives a quality score based on the confidence that both the genotype and the SNP call are correct. Notice that you can also filter on both of these values by using these 2 filters here at the top of the window. Now I'm going to filter this data by using the depth filter, and I'm going to enter 150 so that I'll only see SNPs that occur where the depth of coverage is at least 150. And that takes me down to 7 SNPs. Now, if I wanted to see any of these SNPs in the context of the alignment, I can just double click on them, and that will open up the alignment view and highlight that column for me. To see a graphical representation of this contig, I can go to Contig, Strategy View, and I've already zoomed in here quite a bit so that you can see the regions of high coverage, which are shown on the coverage graph in red. And these are where the amplicons have aligned to the human genome. Down below here, you can see the actual sequences that aligned. And now if I zoom out, you can get a better view of the annotations from the human genome template, which are shown here in this section. If you have further questions about assembling or analyzing ion torrent data using SeqManEngine, please visit our website at DNASTAR.com or contact us at support@DNASTAR.com.